Okay. Hello everybody. I'm Charlie. Deanie, Desi, and Jack are all hopefully in the room with you. And we're group 3. This is our presentation on adult nicotine exposure for Project 1. So, let's just get right into it. We tested 2 flies. The first is Dark 82 strain. This is a genetic control for the A.2.1 fly. And for the sake of the ease of this presentation, we're just going to call it A21. And so this Dark 82 has one mutation. It's a loss of function allele of the DART gene. We'll get more into the DART gene in a little bit. And it essentially codes for a protein essential to apoptosis. Then we have the A21 strain. It's a double mutant strain tested by last year's fall students for fly cure. And essentially the A21 mutation seems to have a growth phenotype and may affect the gene SCAP. SCAP is involved in lipid biosynthesis. It's active in the endoplasmic reticulum membrane, and it's expressed in the fly adult head. It's important to note that this A21 strain has both this mutation on the SCAP gene and the DART mutation. So that's where double mutant strain comes from. All right, moving on to the DART gene. So the DART gene is, you can see right up here, it's right around base pair 17 million, and it's on the plus strand. The mutation is a loss of function allele of the DART gene. And so what does this mean? DART mutants show reduced programmed cell death during their development. So this leads to central nervous system overgrowth. Furthermore, they have melanotic tumors. These are tumors with like heavy deposits of melanin in them. And then they also have various developmental defects. The biggest that we've seen is abnormal wings, both in our research and in our observations. Moving on, there's the SCAP gene. This is right around base pair 6 million. It's on the minus strand. And the SCAP function it's an ER protein, membrane protein that controls the activation of lipid synthesis when levels are low. So it's both sensing when levels are low and then triggering that synthesis elsewhere in the cell. And this mutation affecting flies, SCAP mutants are viable, they can survive. Mammals that are missing SCAP cannot survive. So I thought that was pretty interesting. And the mutation conclusion follows pretty naturally from the function. They show reduced expression of lipid synthesis genes. Great. So this kind of takes us to our central research questions. For the nicotine side of things, how will nicotine exposure affect the behavior and survival of flies? And then for the mutation side, how will mutation impact the survival rate and the behavior of flies? So for our hypotheses, we anticipate that flies that are exposed to nicotine will exhibit more hyperbehavior and not survive as long. And for the mutation side of things, we believe that the A21 flies will have a low survival rate because these flies have more mutations. And these mutations could create less stability in their survival dynamics. As for our justifications, so nicotine-treated flies undergo increased neurological activity, leading to potentially significant symptoms of psychological stress. This disruption in normal cell-to-cell communication can just overall impair neurological function, leading to reduced survival and abnormal behavior over time. So that's how we think nicotine is going to be affecting these 2 strains. As for the mutations themselves, the A21 strain, it contains both the DART and the SCAP mutated genes, as we've discussed, both of which have significant potential to influence behavior and survival of these flies. The DART gene is required for programmed cell death, so a loss of function mutation can lead to the developmental defects that we've discussed earlier. This in turn will most certainly affect their behavior and their survival. At the same time, SCAP helps regulate when cells produce lipids, so disruption can lead to reduced lipid production, whether it be for membranes, growth, you name it, anytime a lipid might be used in cellular processes. So together, it's these mutations are what lead us to the conclusion that altered behavior and survival will be exhibited by these flies. And that's the end of my presentation. This is the method section. Thank you so much, Norma, for letting me do this again. Good luck to the rest of my group mates, and have a great spring break, everyone.